Ladies and gentlemen, welcome to Compass here live on Quotidiano TV today with a very special guest from Australia, from Queensland, Brisbane, Professor Massimo Hilliard. Welcome, Massimo. How are you doing?
Pretty good. Thank you for having me, Radu. Thank you.
What time is it in Brisbane now, if I may ask?
6 p.m. 6 p.m.
So wonderful afternoon.
Few
So it's
hours
winter
away.
time in Australia right now, isn't
It
it?
is. It is. In fact, I almost caught it with the sweater. With the
Yeah, with a sweater, I'm with a t-shirt in warm and sunny Switzerland today.
Yes.
Massimo, all the world had started reading your papers, but let me first introduce you to the audience. You are a professor for molecular and cellular neurobiology at the NHMRC Leadership Fellow, Queensland Brain Institute. And a couple of months ago, you published a remarkably interesting paper in Science, which is quite a prestigious paper for physicians and for scientists. Could fused neurons explain COVID-19's brain fog? That was an absolute breakthrough. I mean, how do you feel about this honor, having published there, and particularly how do you feel the echo in the world of scientists once you published your scientific results?
Yeah, I mean, it was published in Science Advances, not in Science.
Yeah,
yes. But it's good. Yeah. No, there was a good resonance in the field, and we had presented the work also at conference, and the response has been quite strong from colleagues. There was a lot of interest in the phenomenon itself, on what the actual COVID can do to the brain cells. And so our main discovery is that we found this previously unexplained role of COVID in fusing the neurons that it infects. Neurons and glia, which are these other cells that together with neurons make the brains and the nervous system. So the discovery indeed, I think it can provide a third cause of disease in addition to the death of the neuron or the inflammation in the neuron. The neuronal fusion can be a third cause of pathology. And these are not necessarily mutually exclusive. So we can have both, cell death and inflammation and neuronal fusion. But the response was pretty good in the field.
But not looking back, how did you find this out? What inspired you? Were the high number of long COVID cases and the so-called brain fog something where you said yourself and to your colleagues, let's look further to neurons, what might happen on a neuronal level? Right.
So let me give you a bit of a background on how we actually got into this. So we were interested in looking at this molecule called fusogens. And fusogens, their role is to merge cells. And they are present in pretty much every organism. And they do something extremely important. For example, allow the sperm and the egg to fuse. They allow the formation of the placenta in what is called the in situ trophoblast, which is the fusion of thousands of cells that allow the separation of the fetus from the mother. They allow the formation of our muscle cells, the cells they fuse, the formation, the remodeling of the bones, and other processes. And so what we found is that in the nematode C. elegans, which is these tiny worms, these fusogens, they are also expressed in the nervous system. And they do something important. They allow the formation of the dendrites of the neurons, which are this receiving end that allow the neuron to receive information from other neurons. And they're also, and this is the work that we also published, they allow the axon, which is the long process of the neurons, that cable-like structure that allow the neuron to send signals to other neurons. When the axon is broken, the fusogen allow in this species the axon to get back together, to stitch it back together as you would do with an electric cable. They do a very interesting role. But what we found is that we asked how the neurons can regulate the fusogen. What happens if they are deregulated? So if they are expressed in larger amount at the wrong time in the wrong place. And what we found, this is work done in C. elegans, is that when the fusogen are deregulated, they cause neurons to fuse. And that is really the link, because the fusogen, interestingly, they come from viruses. So we asked the question, is it possible that the virus themselves now, the fusogen of the viruses, can cause neurons to fuse, which is exactly the case. And so that's how we really, our work spilled into the viral field for this connection between the fusogen that are present in the organisms and the one that are coming from viruses.
From outside.
And the way and so the way we visualize it is we take these cells in the dish, these are neurons, and we incubate with the virus, and some of the cells have a particular fluorescent marker. And when they fuse, we see the fluorescent marker flowing from one cell to the other. So it's very distinct, very clear.
It's a proof beyond doubt.
Yeah, they do fuse, right? And what we also show is that the reason why they fuse is because the protein that is the spike protein of the COVID, which is indeed a fusogen, the one that everybody now is familiar, that decorates the surface of the COVID virus, becomes now present on the surface of the neuron itself. So the neuron itself now has the spike protein, and this is why it fuses with another neuron.
So the spike, if I may ask, passes the blood-brain barrier, reaches our brain, and then its fusogen component is then interacting with the neuron and is inducing this neural fusion.
Right. So let me just correct that. We
Please.
don't know if the spike itself crossed the blood-brain barrier. That one is not known. Well, we have shown that in the dish, the virus can cause neuron to fuse. And we have shown that in the mouse, if we put ourselves in the brain of the mouse, the fusogen, the neuron fuse. But we do not know if they cross the blood-brain barrier. So that would be, so the virus needs to be present in the brain to be able to exert its effects. Or the molecule itself needs to be in the brain to be able to exert its effects.
Yeah. The neuron theory proposed by Ramon Cajal, or Cajal, I don't know how to pronounce his name.
Yeah, Cajal. Yeah.
Cajal, according to Spanish, I guess, according to which neurons exist as individual cells, has been the foundation of modern neuroscience and has paved the way for our current understanding of how the neuron system develops and functions. Now, what seems now to be different once they start this fusing process?
Right. I think our study, in a way, confirmed the theory of Ramon y Cajal that neurons, they need to function as individual units. Okay. So it's critically important that they stay individual unit, because if they fuse, that can lead to malfunction. And just to bring into the context also, one way of thinking of this is that the neuron has to take care of this fusogen that can come from multiple sources. One sources are the viruses, because the viruses, they can bring the fusogen in the neurons. The other sources could be the fusogen, the endogenous fusogen, the one that we have already in our body that we use for those processes that I mentioned before, which are the placenta, the muscle, that they are normally not expressed in neurons. But if by mistake they become present in neuron, that can cause a damage. And the third possibility, which is also very interesting, is that we carry in our DNA a large amount of viral genes. So 8% of our genome
we inherited.
Is of viral, or 8%. This is an astounding amount of viral DNA. And this DNA that stays in our genome remains dormant. And some of this DNA encodes for fusogens. And so it is possible that in some situation, this dormant DNA become activated. And if they get activated in brains, now the neuron have to face another challenge to how to keep that downregulated. So there are three possible sources: the virus, the endogenous fusogen that we normally use, and the one that we normally do not use, but they stay dormant. That's what the neurons have to challenge. And so going back to the question, Ramon y Cajal, the neuron have to make a task in ensuring that the level of fusogen is kept under check.
But against the background of several symptoms, like the brain fog, we'll talk about potential other symptoms later as well.
Yeah.
Suggests most probably that the spike's fusogen properties is responsible for the fusion and not what we have inherited from our ancestors in terms of DNA information, because brain fog appears frequently now after COVID and has not been something which we were talking about ever since.
Yes, that's correct. So we cannot at the moment precisely tell the cause of brain fog that is caused by fusion, because we know that it occurs, but it will depend on where in the brain fusion occurs and to what extent, what's the magnitude of the fusion. So we can't connect this with the specific symptoms, especially with something so vague as fog. Is not like, for example, having a lack of a particular sense, like olfactory or vision or tact. So this is like more general and a variety of symptoms. But because we know that fusion causes neuronal malfunction, as we have shown, that the neuron, when they fuse, they either start activating simultaneously, or they stop functioning altogether. So no matter what fusion does, seems to be something deleterious. But without knowing exactly what part of the brain at the extent, it's not possible at the moment to associate this particular event of fusion with the specific symptoms. It could be any. So, and also we cannot exclude the fact that is in combination with cell death and inflammation.
Now, one more aspect which I'd like to discuss with you is we have the fusion of this neural fusion, but is initially this mechanism, like many other mechanisms in our body or in nature, one aimed at protection of the neurons? Is this something the neurons start inducing in order to, as a defense mechanism, let's say as a lender of last resort, like a central bank, to put money on the market in case of a crisis, although the risks are very high of some short circuits, which might appear even in an electric system? Is that a defense mechanism?
Right, that's a great
sort
question.
of defense mechanism.
Yeah, that's a great question. I don't think we have a clear answer on that, on why that happens. What we know, though, that at least in the cases that we looked, there doesn't seem to be a benefit at the moment, because, as I mentioned before, when the two neurons fuse, the outcome doesn't seem to be something good for the neuron.
But it could be worse.
Exactly. No, in fact, it could be worse. And it's an important point. So the fact that neuron fuse, it also provides an explanation on the fact that in some neurodegenerative diseases, the toxic aggregates are present in one neuron, and they've seen to spread not only from neuron to the other, but to other brain regions. So it's possible that when neurons fuse, they allow the spreading of toxic aggregates like tau, for example, or alpha-synuclein, from one neuron to the other, to other brain regions. So in this sense, it's not good. But we cannot exclude, and we have not tested this yet, if we have a damaged neuron that fuses with an healthy neuron, if the healthy neuron can provide some beneficial rescue to the damaged neurons. And that is an incredibly interesting question. Is there any benefit, independently on how fusion has happened, for the damaged neuron to fuse with an healthy neuron? So we do not know that.
Once they fusion, they facilitate the passage of tau protein. Did I understand you?
Well, we did not show the tau, but we have shown that when they fuse, very large organelles like mitochondria can move from one cell to the other. So it's very likely, it's almost certain that a lot of other molecules which are much smaller than mitochondria can move as well. Actually, that one, I can't see why, if this large organelle can move, we cannot see movement of a large variety of molecules.
Could also, in case of a prion disease, could also then misfolded proteins, these prions, also travel in this manner?
I think so. I think very much so. I think pretty much a lot of molecules can travel. And that's why you mentioned prion. Prion, it's not known necessarily to cause fusion, but it is possible that can be facilitated, for example, either by viral infection or by, again, the upregulation of some of the endogenous fusogen. And now the spreading becomes much quicker and more efficient because now the neurons are engaging in the fusion. And so I think that is a very likely possibility, for sure.
In one of your articles, you made a brilliant analogy which helps a lot for better understanding of this very complex topic. You say that once fusion takes place, each switch either turns on both the kitchen and the bathroom lights at the same time, or neither of them. It's bad news for the two independent circuits. That's a brilliant example how to explain and facilitate a good understanding of this phenomenon. But could this also have consequences for the way people suffering of this symptom, the way they feel pain? That, for instance, let's say, for instance, they go to the dentist, a usual intervention which is not painful, all of a sudden it's like a huge stroke and they feel a severe pain somewhere in their head. Could that also be the result of such a fusion process? The way we have our emotions, our sensitivity in general, exactly the way we feel pain. Which areas of the way how we interact with our environment might be affected by this neuronal fusion?
Yeah, I think you're absolutely right. So one of the limitation of our study is that we did not connect the fusion to a symptoms, not even in the mouse. And so I think that's an area that is really incredibly exciting to investigate, is what area of the brain, when there is fusion, what are the symptoms that are associated with it? And the prediction is that it can be, as I mentioned before, it can be anything. You can imagine, I think one can imagine that depending on the area and the extent of the area affected, that the variety of the symptoms can vary significantly, because it's all connected to the fact that that is a malfunction, or as you mentioned before, is a shortcut of that particular circuit. And so everything can be reconnected to the concept that when you have fusion, that is causing either a synchrony of activity or a complete abolishment of activity. Neither one of them are going to be positive for the brain. And so depending on where that happens, one can imagine that that's the symptom will correspond to that particular brain that has been affected. In a way, to the same way that when people have stroke, the symptoms can vary depending on what part of the brain has been affected, right? If it's their speech, if it's the movement or balance. I think in a way, it's a similar correlation, what I would imagine. But it's all speculation because we do not have done the experiments to show exactly. And that's why, if I might add, that's why it's really important to, so one of the challenges at the moment is identifying this particular fusion in patients. And the way we thought to proceed is first looking at donated brains of patients that unfortunately have passed away, and being able to identify fusion in patients, and then ideally to be able to identify fusion in a non-invasive manner in patients that are alive, so that one can connect those symptoms. And so we are moving by looking first at the animal model, and then simultaneously to look now in fixed brains, so in post-mortem brains.
This is fascinating. I mean, you've written a milestone in molecular biology and neuroscience. You started with a very generic term, if I may say, brain fog, but we all know that long COVID and COVID is associated with dozens of neurological complications. So maybe, and I wish you good luck and lots of success for you and your team to investigate at which extent further symptoms associated with COVID might be due to this neuronal fusing process. I mean, this would be in medicine an absolute breakthrough.
Yeah, we think so. I think I want to also mention why this we think has been missed is because in patients with COVID, it's already known that in the lung of the patients, what COVID does makes cells to fuse, which it contribute to the pathology. And so it's easy to see in the lung of the patients because the cells are all round. And when they fuse, it's easier to see this large, what we call syncytia, which are the fusion of multiple cells.
Is this related? Sorry.
No, go ahead.
I read some papers in radiology because I also intend to look at the so-called mosaical pattern which a radiologist observed after a CT in the lungs of COVID patients, which was formed by amyloid fibrils. Forming these mosaical patterns, but I still don't know yet. Look, I'm not a physician. At which extent the amyloids, which are also a molecular fusing process on their way to nucleation, if these amyloid fibrils facilitate or not this fusing process in the lungs you're talking about.
So I don't think I'm not sure if fusion will be visible at this level. It will be probably more visible at the pathological analysis because they need to look at the actual tissue to be able to see that. So it's already one level. It is possible that there is an effect that I'm not aware of, but what I was discussing is the fact that looking at the tissue, you will be able to determine that there has been some fusion. And now in the lung, this is a little bit easier for the morphology of the cells, which are nice and round or have hexagonal forms, right? The reason why we think this has been difficult to see in the brain is for the very nature of the neuronal cell, which have all these long processes. And so the fusion does not happen at the level of the soma, what we call the soma, it's where the nucleus or the main part of the neuron, but it can happen a very long distance. We have shown that it can happen when the neurites, which is the neuronal processes, get in touch with each other. And so from a neuropathological analysis, it will not be seen. It will be extremely difficult to detect. And that's why we think this has been missed from the neuropathological analysis, because it's extremely challenging to find. And we can do it because we can manipulate the cell in culture. We can put fluorescent markers in. Everything is the cell are alive. So we are in a very privileged position to be able to detect the fusion event. But from the tissue collected from patients, it's a very different story. So I think that's what has probably created a limit in this understanding of this process.
Would also autoimmune processes induce the fusion mechanism?
Right. I think it's a great question. I want to come back to the fusogen.
Yes.
So yeah. So the question you're asking is extremely interesting. So two cells that are close to each other, there is a thermodynamic barrier for them to fuse. So they are born to stay separate, to get close to each other, to interact with each other, but to stay separate. So to be able for them to fuse, you need to have this fascinating molecule called fusogen that allow to reduce this thermodynamic barrier and allow the membrane to engage in fusion. And so and just to give you an example, one that we know very well, which is again in the nematode C. elegans, is this fusogen called EFF1, which function as, we call it trimer. So there are two molecules on one side of the cell and one molecule on the other. And when they get together, they in touch, they change conformation, they flip like a spring, and they bring the two membranes close together. And now the reason why I mention this is because it is possible that inflammation or autoimmune diseases can trigger the expression of fusogens. And because that's what you need to be able to have the cell to fuse. And so I don't think we have a full understanding on what are the possible causes that can create an environment in which now the fusogen become expressed in neurons, and then as such, the neurons now become capable of undergoing fusion.
Have such cell fusions been observed in the context of prions, or in this case of misfolded prions?
Not that I know of, but I think it's a field that it's worth investigating. I'm not aware that the prion itself can cause fusion, but we know that it can spread. And so it is possible, and again, this is full speculation, that it's possible there are some elements that either there is an expression or overexpression of dormant fusogen or the endogenous fusogen upon the prion or the fibril formation that then cause the fusion, and then the prion can spread from one cell to the other. But the prion itself being fusogenic, I'm not aware of. And yeah.
The reason why I'm asking, I mean, the prion is like a new term, thanks to Professor Prusina. He even received 1997 the Nobel Prize for his discovery. But looking back in the history of medicine, back to 1978, Daniel Gajdusek and his colleagues, they published a paper, Cell Fusion Induced by Scrapie and Creutzfeldt-Jakob Virus Infected Brain Preparations. Now, they use the old terminology, and these days it was the slow virus and not the prion, but we know what we are talking about. So in their case, the slow virus was able to induce Scrapie is today a prion disease. For Gajdusek, it was a slow virus disease. And he described that Scrapie theoretically could induce this neural fusion. Then theoretically, the synonym or the modern term should at least facilitate the same fusion. It's purely speculative what I'm asking. What they've observed 78 should then theoretically apply also to prions.
Yeah, and again, I think it's a field that is worth investigating. I'm not sure if there is evidence that, because to be able to be a fusogen, it needs to fulfill several criteria. That needs to be present, there needs to be sufficient to cause fusion. So there are several criteria. And so it's possible that they need other factors for fusion to take place. But it's interesting because the spike protein itself can be, if it's cleaved in a specific, I think the spike protein itself is not necessarily forms amyloid fibrils, but if it's cleaved in a specific way, some of the peptides of the spike protein can become themselves, they can form fibrils, not being fusogenic, but being fibrils themselves. So it's a different role. And I don't think that they've been looked in the brain, but I think in the blood, they're known to form these aggregates.
But could it be like, to compete in easier terms for the audience, and also for my better understanding, could we say that it is like a two-component glue? Like, according partly to the second component, our genetical susceptibility, some have more or less of the second component, and then they get in touch with the first component glue, which could be an amyloid enhancing factor encoded in the spike. When the two components of the glue come together, they form more or less or not at all these amyloid fibrils.
Yeah, I don't have an answer because I don't know enough about that, but I would imagine that to fulfill the criteria of being fusogenic, you need to be able to express that in cells and showing that they undergo fusion. Okay. And so, and I don't know if that's the case. What I would be, so that's even the spike protein, we know that the spike protein needs a receptor. So when the virus that is covered with the spike encounters a cell, it needs to have a receptor called the angiotensin converting enzyme 2 or HACE2 that needs to be present on the receiving cells to allow fusion to take place, to allow the virus to enter our cells. And we have shown that even if the neuron have the spike protein, they need to have the receptor on the adhesion cells for fusion to take place. So it's not necessarily one molecule. You need to have two molecules for this to function. Now, I think that it's possible the prion is causing somehow the expression of fusogens on the cells, which in turn now the cells can undergo fusion. That would be probably what I would imagine. But again, that's really outside my field.
Something to investigate.
Yeah, it's something to investigate.
Do we know anything now, since we're approaching the end of this very interesting interview, do we know anything about the ongoing life of such neurons once they fused? Is their life expectancy shorter, or is it a normal process?
Yeah, that's a great question. I think at the moment we think that in the majority of cases, for the time that we keep the neurons alive, and again, these are in the dish, the neuron seems to be doing fine. So again, their function is altered in the sense that they either function together or they do not, but they seem to be fine. So we don't think there is an association associated with cell death. But of course, in the long term, we do not know if that's the case. What we know from the study in the nematode, in the C. elegans worm, we know that when the neuron fuse, the animals stay alive and the neuron still function properly for the life of the animal until the animal becomes adult. And this is a short time, but it's a long time in the life of a worm, but it's a short time for us. So it's days. But they can still function, and we do not see cell death appearing. So my tendency would be that probably it's something that can happen, but at the moment we think that the cell can stay intact for quite a long time, which distinguishes them as a mechanism from the cell death.
Guy Dussek, I'd like to quote two sentences from the end of his paper, which I mentioned before, from 1978, related to neuronal fusions after scrapie, a prion disease inoculation. He says that these observations suggest that fusion induction may be a property common to the agents of subacute spongiform encephalopathies. The ubiquity of some enveloped viruses poses constraints of specificity of cell fusions as an assay for the spongiform encephalopathies. I still need to practice this name. Nevertheless, it will be of great interest to test various preparations from tissue of patients with Creutzfeldt-Jakob disease or Kuru, as well as brains from patients with other neurological and non-neurological disease. So it's quite amazing, isn't it? They even calculated the speed of this fusion and published the fusion index. It looks like, to me, it looks like math, but apparently this mechanism is still something which will keep scientists like you busy in future as well.
Yes, yes.
Oh, there will be the first speed trap for the neuronal fusion. Professor Hilliard, thank you very, very much for having joined me today. I really appreciated your effort and the brilliant way you described this neural fusion. Wish you once more and your colleagues lots of success in investigating for the benefit of all of us, for our health, this very important matter. Greetings to Australia and have a nice evening.
Thank you for having me, Radu. I really appreciate it. I enjoyed the interview. Thank you for your insightful questions.
It was my pleasure. Bye-bye.
Bye-bye.